Hi there. So in this video, we are going to be going through the tutorial for biotechnology. Just a reminder, before you watch this, you should actually watch the lecture recordings for biotechnology first. This is just, again, a summary, a condensed version of what you should know. So here, let's start with the short answer questions. The gel on the right was run after bacterial plasmid was digested using restriction enzymes. Indicate following on the gel. Okay, so this is called gel electrophoresis. Let me write that down for you. Gel electrophoresis. Okay, so in gel electrophoresis, you basically would load your DNA sample in the wells here. And then the gel is basically made of like a polymer, and it's kind of like a mishmash for the DNA to kind of weave through. And you would apply a current to push the DNA from one side to the other. Okay, so you would have to hook this gel. You actually would put the gel in like a buffer solution, and then you would apply electric current to it. So over here, these slots, that's where you would load your DNA sample. Okay, so loading well. Okay, that's where the DNA sample goes. And then what's going to happen is we are going to hook this side up to the negative electrode. Okay, so that's the negative side. And then this side would be the positive. Okay, and then you're going to turn on the current and it will go from here to here. So this is the direction of current. Okay, I means current. And why would that push the DNA from one side to the other? Well, remember the DNA backbone is a sugar phosphate backbone. Okay, and phosphate, as you remember, carries a negative charge. So when you load the DNA sample here and then you apply the negative current, it will push the negatively charged DNA towards the positive. Okay, so you will run this for, maybe 30 minutes, maybe 40 minutes, depending on the application. You don't want to run it for too long though. Okay, if you run it for 3 or 4 hours, it's possible that all the DNA would eventually exit from the bottom of the gel, and you won't be able to see anything at all. Okay, so that takes care of question A and B. And then the next question is the location of the largest band. Okay, so the band that are really big, the DNA. So just to refresh your memory, right? Like there is the plasmid within the bacteria. Plasmid is basically circular DNA. So you take the plasmid and then you cut it up into like pieces. So you will have a long piece, maybe shorter pieces like that. So the pieces that are really short, they are going to be able to weave through these mishmash much easier. Okay, so they are going to be able to travel further down your gel. So if you're looking for the largest band, that is the band that corresponds to the longest DNA, that would be the one that didn't really go anywhere because it's too big for it to travel down the gel. Okay, so that's your largest band. So by the same logic, the smallest band would be the one that is able to travel the furthest. Okay, so that's 1 sample, 2 samples, 3 samples, 4 samples. Over here, you will see on sample number 3, we have our shortest band right there. Shortest band. Okay. So next, we're going to have to identify the number of cuts that were made on the plasmid, and then the question asks, will the answer be different if the DNA was linear? Okay, so let's take a look at here. Okay, let's try to create another plasmid. Here is a plasmid. Okay, let's say I cut it once. Right? If I cut it once, after 1 cut, right? If you think about a rubber band, after you cut it once, it's just going to be still 1 strand, right? Okay. So what if I have 2 cuts? Okay, 1 cut here, 2 cuts here. If I cut it twice, then I would have 1 band here and then a second band, right? Okay. So here it says plasmid are being loaded. In other words, the number of bands I see on the gel is going to tell you how many cuts was used. So the fact that we have 4 bands here in the first sample, that means there's going to be 4 cuts in this sample. Okay, so don't believe me? Let's take a look again. We have the circular plasmid. If I cut it 1, 2, 3, 4 times, then I will have fragment number 1, fragment number 2, 3, and 4. So that's how I get the 4 cuts. Okay, so for the next one is 3 cuts, right? 5 cuts and 4 cuts. Now, this second part is important. Will the answer be different if linear DNA was used instead? Okay, so let's say instead of starting with a circle, I start with a linear DNA. So if I cut it once, then that one cut actually will produce 2 bands. Okay, so in other words, if you see 4 bands, in your sample, okay, and it was linear DNA that you're using, then actually it would be just 3 cuts. Okay, so just to summarize for you, I guess, if you have plasmid, right? So number of cuts equals to number of bands on the gel. If you have linear DNA, the number of cuts would produce let's say you have like n number of cuts, right? We cut it n times, then you would have n plus 1 number of bands. Okay, whereas here n number of cuts will equals to n number of bands. So 4 cuts will give you 4 bands. 4 cuts on linear DNA, that will give you 4 plus 1, which is 5 bands. Okay. All right, let's get back to the study guide. And let's take a look at the next question here. What is the reason for using tag polymerase? Actually, you know what? Let's go to here. This one I can actually make more space. Okay, what is the reason for using tag polymerase? So in PCR, what is PCR, right? Well, PCR is basically it stands for polymerase chain reaction. Okay, is a technique used for cloning DNA sequence. Okay, so let's say this is the long strand of DNA. Okay, long strand of DNA, and then double-stranded, and I want to make a lot of copies of this particular section. Okay, so what you can do is you will design primers that will flank the gene that you're trying to clone, and then using PCR, you will be able to make multiple copies of it. Okay, so in the process of PCR, right, so PCR involves heating DNA to very high temperature. Okay, so like maybe 60 degrees, 70 degrees, depending on you know, the section that you're trying to clone. So at those temperature, regular polymerase is going to denature. Okay, so regular polymerase, like the ones that find in our body, for example, they will denature at these high temperatures. Okay, so what's so special about the tag polymerase? Well, tag polymerase is extracted from bacteria that live in hot spring. Okay, So these polymerase are adapted to operate at a high temperature, right? These hot springs can sometimes reach the hundreds of degrees. And so this particular protein will not denature at high temperature, making it ideal for PCR, okay? When I say extract, like, I don't mean like we actually gather these bacteria and then grow them or something, right? These things are nowadays being produced through biotechnology anyways. Okay, so what are the 3 major steps in PCR? So I posted some videos online for you to actually see the animations of how it works. But to summarize it very briefly, right? How that basically works is first step is denature. Okay, denature. So we have our DNA strand here. Let me try to move this down here. We have our DNA, which are stuck together, right, because of the hydrogen bondings. So the denature steps, we would heat up the DNA. That would cause the DNA strands to separate. Okay. And then the second step is we will cool the temperature a little bit, right, for the next step, which is called anneal. Okay, so this anneal step allows DNA primer to bind to template DNA. Okay, so these are the primers. The primers tells you, it tells the process which section to copy. Okay, remember these are DNA, right? So it's like AAT, CG, blah blah blah, a bunch of nucleotides. So by creating complementary primers, you can decide which section to clone. Okay, if I have a primer that goes from here to here, then we're going to clone this entire section. Okay, so the primer marks the beginning and the end of where we want to clone. Okay, when I say clone, I mean make many copies of it, right? Okay, and then number 3, right? We're gonna lower the temperature even more, so it allows the elongation, the actual copying, to take place. So the polymerase, the tag polymerase, copies DNA. Okay, so in actuality, what happens is once you cool the temperature a little bit, then the tag polymerase will come and then it will copy this section, and then it will copy this section. So after one round of copying, right, you will have the original DNA, okay, and then you would have a copy of the section that you want to make. Okay, so then you would do it again. You would denature these things. You would lower the temperature a little bit afterwards. So then the primer will bind here and here, but then the primer will also be able to bind to the cloned copy. And then what's going to happen next is, I'm going to use a different color to show you. What's going to happen next is the tag polymerase will copy here, and then same thing here and here. Okay, so after 2 rounds, you would have a total of 4 copies, and then it just goes exponentially. Okay, so that's how PCR works. And again, for the more detailed explanation, please go back and look at the lecture recordings and definitely watch the animations that I posted. Okay, so next question, what are restriction enzymes? Okay, so restriction enzymes are normally found in bacteria. Okay, so we'll say naturally found in bacteria as a defense mechanism. Against foreign DNA. Okay, so in a bacteria, right, they would have their own DNA. Some of them would be plasmids, but their DNA are going to be methylated. Okay, methylated. That means there are like CH3 on them, on the DNA. Okay, so when it's methylated, it kind of tells the bacteria that, these are my DNA. Okay, and then you would have these restriction enzymes that are found in the bacteria. Okay, so sometimes bacteria, they might pick up DNA from the surrounding environment. Okay, so let's say there's a DNA. Excuse me. Let's say there's a DNA that got picked up by this bacteria from outside. So this is like foreign DNA. Okay, so the foreign DNA are not methylated; they don't have the CH3 on it, and then the restriction enzymes will now come and cut this out. Right, so it's not going to affect the bacteria. Okay, now how do the restriction enzymes work? I'm gonna need more space for this. So restriction enzymes, they work by cutting at certain recognition site. Okay, so let me write that down for you. Restriction enzyme RE cuts at certain recognition site. Recognition sites. Okay, and each restriction enzymes are going to have their own unique recognition site. Okay? There are many restriction enzymes from various bacteria. Okay, so they are named usually after the bacteria that is found in. For example, there's something called E-core-R1, which is extracted from E. coli. Okay, so different bacteria have different class of restriction enzymes, and they all sort of have their unique recognition sites. But what's common between all these recognition sites is that the sites are all palindromic sequences. Okay, palindromic sequences. Okay, in other words, it reads the same forward as it does backwards. So let me give you an example here. Give you an example. Let's say we have GAATTC. Okay, so this is like 5' to 3'. And then on the other end, if we use complementary rule, it will be like CTT, because G pairs with C, A pairs with T, and then we have AAG. So this is a palindromic sequence. Okay, palindromic sequence, because if you read it forward, GAATTC is the exact same when you read it backwards, GAATTC. Okay, so if you treat it with a restriction enzyme, okay, let's say we treat it with something called like E-core-R1. Okay, that's a 1. I know it looks like an I, but it's really like a 1, and is extracted from E. coli. What happens is anytime the E-core-R1 see this sequence in any DNA, it will cut it like this. Okay, so afterwards we're gonna have G and then C T T A A plus the other strand is gonna be AATTC and then with a G. Okay, so it's gonna look like this. Right, so these are called sticky ends. Sticky ends. Okay, because this part will be able to stick back to here, right? Okay, from this end to this end, because they're complementary and they're able to, rejoin together if they find each other. Again, that's going to be important for what we discuss later on. Okay, but that's not the only way this could happen. Let me give you another example of another palindromic sequence. So let's say CCCGGG. Okay, so on the other strand, it will be GGGCCC. So you see, reading forward and backwards is the same sequence. So let's say I treat it with another restriction enzyme. We're going to go like small one. Okay, it's another restriction enzyme from another bacteria. And anytime this restriction enzymes see the CCCGGG, it is going to cut like that. Okay, and then we're going to get CCCGGG plus GGGCCC. Okay, so instead of having the sticky ends, right, these are not gonna, be able to come back together, and we call these blunt ends. blunt ends. Okay, so for the purpose of like a lot of the application we use in biotech, we want to create sticky ends. Okay, so that's what restriction enzymes are. Okay, next one. What are STRs? Okay, or short tandem repeats. Okay, so let me just give you like the basic definition first. These are basically stretches of DNA that contains repeating units of 2 to 7 nucleotides. Okay, so for example, right, you might see like ACG5. Okay, what does ACG5 means? It means on the chromosome, on the DNA, oops, that's not a very nice looking chromosome. There we go. On the chromosome at a specific location, right? If you have an ACG5, it means at that location it's going to be like ACG, 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 ACG. Okay, so it's going to be repeated 5 times. 1, 2, 3, 4, 5. Okay. And then, you know, on another person, they might have like ACG3. So instead of repeated 5 times, it would just be ACG, ACG, ACG 3 times. Okay. So these are short as in 2 to 7 nucleotides. A tandem means one after the other, and they are repeats. Okay. So in different people, the number of repeats might be different. Okay. Some people might have more repeats, other might have less repeats. Okay, if you are related, right? Like let's say in this location, right? Like if your parents is ACG5, right, on both chromosomes, then you are likely to have the same number of tandem repeats as them. Okay, it's possible that you might have like some extra or missing, like you might be like an ACG4, ACG5. And the reason is because whenever there are like a lot of these repeats, these tandems, the polymerase actually slips when it's making the copy. Okay, and it basically makes a mistake. Right? It might add like an extra repeat or like a missing repeat, and that's how we come up with these variations in the number of repeats in individuals. Okay, imagine, right? Like if I ask you to copy the sentence, you know, how are you today? You can copy that, probably not going to make a mistake. Okay, but if I ask you to copy this, okay, like copy that like 10 times, right? You have to pay attention to how many T's you're copying, right? Okay, you might, if you're not careful, you might have an extra T, or if you have a missing T or something. And that's kind of what these tandems are. Okay, the polymerase kind of, you know, make errors, and it creates extra or missing repeats. Usually it's not a problem. Okay, sometimes it could be a problem. In the lecture I talk about Huntington disease. There's like a critical number of repeats, beyond which, if you have too much of it, then you know you're likely to have the conditions. But usually these tandem repeats are not going to cause any problem in the body, right? Too much or too little of it's like, it's usually not a cause for concern. Okay, so that's what STRs are. Okay, next question. What are RFLP? Okay, so RFLP stands for restriction enzyme. Restriction fragments rather. Restriction fragment length polymorphism. Okay, I'm just gonna erase some of these repeats. Okay, restriction fragment length polymorphism. Okay, so these are fragments of DNA created by cutting them with restriction enzymes, and the lengths of the cuts of the fragments that you create could be different between individuals. Okay, so that difference is called polymorphism. Polymorphism just means that subtle differences between two people's DNA. So for example, if you are ACG5 and then your brother or sister is ACG4, that's a polymorphism. Okay, so what exactly is it, and how do we use it? Well, again, I know I keep on saying this all the time. Watch the lecture recording for the full explanation, but I'm gonna try to explain this to you with one example. Okay, so let's say we have a strand of DNA from individual number 1. Okay, we'll say person A. Okay, so let's say DNA sequence, and then we have like a C, A, T, G. Just give me a second. And then I'm gonna throw in like a C, C, C. I'm gonna need more space for this. C, 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 G, G, G. Okay, almost there. I'm just gonna make up a few more things. Okay, let's say there's a sequence like this. Okay, and then I'm gonna just write the other half for you. Okay. C, 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 A, T. Okay. So let's say there is this sequence in person A. Okay. So what happens is, let's say I treat this with small 1. Okay. You remember small 1 from the above example? So small 1 will look for the palindromic sequence CCCGGG. Right? CCCGGG. And then it's going to cut it right there, creating a blunt end. Okay. So afterwards, you are going to have 2 segments. Okay, I'm just going to copy this, see if it's going to work. Sort of worked. Okay, so there is going to be one segment, and then we're going to have another segment right here, right there. Okay, so if I cut this segment from person A, and then I run this on a gel, on a electrophoresis gel, okay, like this. Okay, let's say I load the samples. This is the digested sample, right? And then I load it here. I am going to see 2 bands. Okay, 2 bands. They're similar size, so they might be at the similar position. Okay, so 2 bands. Okay, now let's say we have person number 2, person B. Okay, and at this exact same location, they would have the same sequence. Okay, I'm going to put it right here. But let's say in person B here, there is a point mutation, a neutral mutation. So instead of like a G here, it got mutated to, let's say, like a T, and then the other side would be A, right? So like a neutral mutation. Just cause, okay, just because people are different, right? So that's a polymorphism. Okay, instead of GC, it's going to be TA. So with the introduction of a TA, you can see the palindromic sequence that was there in this person is not present in person B. So now if you treat person B with small 1, what's going to happen? Well, what's going to happen is nothing's going to happen, because the palindromic sequence that small 1 recognized is no longer there. So if you treat it, it would just be the exact same thing. Okay, nothing happens, no cuts. Okay, because again, the palindromic sequence recognized by this is no longer present. So if you load this sample, right, you're just going to have 1 band. Okay, so I can, based on the number of bands I get after I treating the sample with small 1, I can distinguish whether the sample is from person A or person B. Okay, so this could be useful, like, I don't know, from like a crime scene or something. Let's say you find a DNA sample in the crime scene. Okay, is it person A sample or person B sample? Well, then I can treat it with, like, treat the DNA sample with small 1. And then after I load it, I see 2 bands and I know it's from person A. If I only see 1 band, then I know it's from person B. Okay, of course, like this is the principle of it, right? The actual thing is much more complicated. and you're going to need to have multiple restrictions enzymes, and you will have to identify these RFLP sites. But basically, in a nutshell, that's what it is. Okay. Next, long answer questions. Okay, describe how a gene can be inserted into a plasmid. All right. So again, just to refresh your memory on what a plasmid is. Okay, so a plasmid is a circular piece of DNA. Okay, plasmid. It's circular DNA. Okay. So what happens is, let's say I have a gene here. Let's say I have a gene here that I want to put in the plasmid. And then I can feed the plasmid to a bacteria, and then the bacteria would express the gene, right? Okay, so how do I put this gene into the plasmid? So assuming this gene is already cloned using PCR, right? So one of the criteria you want to have before inserting the gene is to make sure that the plasmid will have the same recognition site for restriction enzyme as you would on the gene. Okay, so we're just gonna use our previous example. Okay, so in our previous example, our restriction enzyme, for example, there was the GAA-TTC, right? So I'm gonna put it here. I'm gonna put GAA-TTC, and then on the other end is CTT-AAG. Okay, so, and then this is gonna be the gene of interest. Gene of interest. That's the one you wanna pop into the plasmid. TTC, CTT-AAG. And then the circular plasmid, let's make it a little bit bigger. Okay. And then we would have to have the same thing. Okay. So I'm just gonna copy it. GAA-TTC, CTT-AAG. Okay. So long story short, again, full explanation, watch the recording. Must be sick of hearing me saying that. So if I treat this with, say, E core R1, what's going to happen is it will cut it like this. Okay, because we saw that earlier, right? Okay, and then if I treat this also with E core R1, then it's going to cut it again like that. Okay, they cut in predictable ways. Okay, so I'm going to cut it like that, and over here it is going to create the same cut like that, right? So what is it going to look like now? What is it going to look like after that? I need more space. Never have enough space on this thing. Okay, so this one is going to pop the circle open. Okay, and then on one side it would have a G and then CTTAA. On the other side, we would have a G, CTTAA, because you cut it open, right? So now I have a sticky end right here, and then I have another sticky end right here. Okay. And then this gene, what's going to happen after you treat it with E core R1? Okay, after you treat it with E core R1. Okay, so E core R1, what's going to happen is, let me see if I can copy this so I don't have to redraw the whole thing. Okay, so what's going to happen is, sorry, it's like all over the place. Maybe I put it over here. Okay, E core R1. What's going to happen is this will be gone. Okay, and then this will be gone, right? Because it was cut away, right? And you will have a sticky end here. Okay, and then you will have a sticky end that goes right here. Okay. And then you mix these things together. You can mix these things together. And what's going to happen is, let's take a look at this sticky end on the gene. Okay, this sticky end is AATT, right? Which is perfectly complementary to the sticky end over here. TTAA. And then similarly over here, TTAA is perfectly complementary to the sticky end over here on the plasmid, AATT. Right? So essentially, what's going to happen is this. This thing here, I don't think this is going to work too well, but I'll try. This thing here is going to basically fit right in here. Okay? With the complementary strand binding to the sticky end. Right? So in the end, maybe I just take that away. It looks kind of silly. So in the end, you will have your plasmid, and then you will have your gene of interest inside. Okay, the gene of interest. Okay, because once again, this part here will stick to here, and this part here will stick to here. Okay, the key is to cut the plasmid and the gene of interest with the same restriction enzymes. That way, they're going to have the same sticky end, and then they will be able to bind to each other. Of course, you have to add a little bit of ligase to glue it back together. Right? Remember, DNA ligase create that a bond, right, to link the DNA together. So that's what's going to happen. That's what you need to do. Okay, no use creating the blunt ends is not going to work. Okay? For you to insert the gene, you would have to do sticky ends. Okay, so what are you going to do with this plasmid with the gene in it? Well, you can feed it to a bacteria, and then the bacteria will transcribe and translate this gene. Okay, maybe this gene codes for insulin, for example. Then the bacteria can mass produce insulin, which you can then extract afterwards. Okay, that is that. Okay, next one. Describe how to determine if bacteria have accepted the plasmid that you transform into them. Transform means to basically feed the plasmid to the bacteria. Now, we can't really physically see these things, okay? Even if we put the bacteria under the microscope, you won't be able to see whether the plasmid has been accepted, okay? So usually what happens is on the plasmid that you're going to feed the bacteria, okay? You're gonna have like a antibiotic resistant gene in it. Okay, so let's say we have like TETR. Okay, so like it's resistant to tetracycline. Okay, which is an antibiotic. Okay, so then what's gonna happen is you're gonna insert the gene of interest using the technique I mentioned above. And then you're gonna feed it to the bacteria. Okay. So how do you feed them? Like you mix them together in a solution, and then sometimes you can shock them with electricity, and then they will take them up. Okay, that's one way of doing it. And then what happens is you're gonna grow those bacteria on a culture plate. Okay, so on the culture plate, this is a plate. Culture with the nutrient and stuff. Okay, and then you're gonna add some tetracycline in it. Okay, so the bacteria, if they're able to grow, right, all these bacteria that are growing in the presence of tetracycline means they have received the plasmid. Okay, because that plasmid, in addition to having the gene of interest, also has the gene that resists tetracycline, right? So if the bacteria did not take up this plasmid at all, then they would have died, right? They would have died. Okay. Now, what if you have a culture? Okay, this is important. What if you have a culture and then you have tetracycline in it? Okay. And then you put the bacteria on, and then after a day or something, you look at it. There's no bacteria at all. No growth. Okay. What does it mean? Well, does that mean the bacteria did not take up the plasmid? Okay. Could be. Okay. If none of them takes up the plasmid, then none of them would have the tetracycline resistant gene, and then all of them would die, right? But then you don't really know. Maybe it's not because they didn't take up the plasmid. Maybe you accidentally kill them all. during the transformation process. When you try to shock them with electricity to help them pick up the plasmid, maybe you use too much voltage or something and you kill the bacteria. Okay, maybe you know there was something wrong with your culture plate. Right? So how do you know? So usually we're gonna have to have a control. We're gonna have a control plate. Okay, so this plate is gonna be just the culture. Okay. With the nutrients. Nutrient is called like LB, okay? LB is the nutrients, okay? So with nutrients, but no antibiotics. So you would take this bacteria, which you have fed the plasmid to, you would play them here and you would play them here, okay? So if you see that they grow over here in the control without the antibiotic, but then they do not grow in the one with the antibiotics, then it tells you something was wrong with the transformation process, right? None of them take up the plasmid. It's really unlikely that none of them take up the plasmid, right? Okay. So this control tells you that the bacteria themselves, nothing's wrong with them. They're able to grow. Okay. If both the control and the culture with the tetracycline have no growth, then you have to do some investigation. Okay. Maybe all the bacteria die somehow. Okay. For some other reason, not related to transformation. Okay, so that's why we need to have a control. Okay, so if you see growth in the control, right, a lot of growth, right, and then maybe you see some growth here, right, so some growth, right, which would make sense because only certain percentage would take up the plasmid, right? Okay, so that's how you know whether the bacteria has accepted the plasmid or not. It's by growing them in antibiotics. Okay, explain how STR and RFLP can be used to compare a DNA sample. I mean, we sort of talked about it already in the above example, and there is much more detailed explanation you can see in the lecture recording, so I'm not going to go through that again. Next one, what are some challenges related to the use of gene therapy? Okay, so gene therapy can be used for various reasons. For example, you can replace a missing gene that's defective. You can knock out a gene that's too active, for example, right? But the challenge with gene therapy can be summarized in a few points. For example, target delivery. Okay, that is the concern, right? How can you ensure that the gene you are giving to the person is going to end up where you want it to be? Okay, so let's say on chromosome 4 or something, there is a defective copy of the gene at this particular location. So the idea is to deliver like a functional gene to replace that faulty gene. But how can you ensure the gene is going to go here and not like here or here? Because randomly inserting genes in random locations could have very bad ramifications, right? You can destroy or interrupt other gene functions, and that's not a good thing. Okay. Another thing is immune response. Okay. Because after all, you're introducing foreign material. Sometimes you are delivering it with like a viral vector, right? Your immune system might not like that. Okay. And I briefly mentioned already, right? Like interaction with other genes, right? We call these like off-target effects, right? What if this gene that you're inserting cause another gene to become malfunction, right? Then we have other problems, right? And cost is a big issue, okay? Because these gene therapies tend to be very personalized, then it's going to actually, be quite expensive, right? Because it's almost kind of like a tailored type of therapy, of medicine. Okay. Next one, PCR requires 2 DNA primers to be used, one that is complementary to 5' region of the target areas on the one strand and the other complementary to the 5' region on the opposite strand. Predict what would happen if only one primer were available. Okay, so just to quickly remind you again, let's say this is your DNA, right? You would have a primer here, you would have a primer here, and then you would copy the DNA like this. So after one round, you would have 2 copies. Okay, so 2 copies. 1 cycle of PCR, you will have 2 copies. Okay, so I'm just going to draw this original copy and then the cloned copy. And then if you have another set of primer like this, right? Then you would copy and copy like this. So after another second cycle, so second cycle, right? I should say this is first cycle, you would have 4 copies. 1, 2, 3, 4. Okay. And then if you do another cycle, you would have like 8 copies. Okay. So PCR is exponential. Okay. So like if you start with 1 copy, then you're gonna have 2 copy, 4 copy, 8 copy, 16 copy, so on and so forth. So if you have like 10 cycles, it will be 2 to the exponent 10, which will give you 1,024 copies. Okay. So what happens if you only use 1 primer? Okay. What's gonna happen? Well, if you only use 1 primer, so I'm gonna erase this. You're gonna just use 1 primer. Then you will copy this strand, but not this strand. Okay, so you will have the original strand, and then you will just have 1 side of the of this strand. Okay, so like you would have like 2 copies. Okay, and then what's going to happen is you're going to do it again. So here and here. Right? And now you're just gonna make 1 copy and then 1 copy. Okay, so after another cycle, you would have 3 copies. Okay, so if only one primer is used, then the cloning is linear as opposed to exponential. So it's like 1 copy becomes 2, becomes 3, 4, 5, 6, ... So if you do 10 cycles, you would only have 10 copies as opposed to thousands of copies. Okay. Explain how gel electrophoresis work. Just look at the, again, the lecture recording. It's much more detail. And again, there is a video that shows you in real life how it works. And I sort of explained it at the beginning already, so I'm not going to go through that again. Next one, give 3 examples of how gene therapy works. So I said you can replace non-functional gene. You can suppress an overly active gene. Okay, so if there's a gene that causes your cell to divide uncontrollably in some cancer, you might want to suppress it. Okay, you can also use it to modify the clinical course of a condition. Okay, so maybe you can introduce a gene to like the white cells and then make them recognize, certain targets on a cancer cell, and now they would attack it. Okay, so these are 3 examples of how you can use gene therapy. Okay, let's move on to the hard stuff now. Here's some application question. The number 7 right here. The recognition site for BAMH1 and BCL1 is shown below. Okay, so again, you can see the palindromic sequence, and then they cut it, creating sticky ends. So if the following DNA was cut with BAMH1, how many DNA fragments would you expect? Okay, so it's quite simple. BAMH is going to recognize GGA-TTC. So I'm going to scan this sequence and look for GGA-TTC. So I see GGA-TTC right here. Okay, and you want to see if there are other GGA TTC. I don't think so. I think those are the only ones. So if you treat this with BAMH, it's going to cut it like that. Okay, so how many fragments do you expect? 1 fragment, 2 fragments. if you treat the DNA fragment produced in part A here with BCL1, what results are expected? Okay, so the fragments produced in part A, right? So over here, if you treat it with BCL, so you're looking for TGA, TCA. There's no TGA, TCA in this part. And then if I look over here, TGA, TCA is right here. Okay. So then the BCL is going to cut it like that. Okay. So this is the original fragment. Okay. And then if you treat it with BAMH1, you're going to get a short one and then a long one, right? Together it makes up this one, right? And then if you treat this thing, this whole thing, so let me put a cut here, put the cut, the first cut, okay? And then if you treat this with BCL1, what's going to happen is nothing's going to happen to the short one. And then you're going to cut this one like that, and then you will have 3 fragments. Okay, so after the second cut, you will have 3 fragments. Where's the question? Right here, 3 fragments. Does that make sense? Any question? Oh, sorry, I forgot I'm doing a recording. I thought I was doing a live section. And in live session, I always ask you, do you have any questions? If you do have any questions, of course, please email me. Okay, so that's that. Next one. Suppose you run DNA sample containing the above DNA segment, like this one, on an agarose gel with the following treatment. Okay, so we're gonna draw 4 lanes. Let's draw 4 lanes. Let's see what happens. 2, 3, 4. Okay. There's your gel. Okay, this is the negative side. This is the positive side. This is where you load the sample. Okay, so lane 1, untreated, just the DNA sample alone. So in other words, it's just this sequence with no cuts. So if you load it, this is pretty big. So maybe it's just gonna stay close to the well. It's not gonna go migrate very far. Lane number 2. Okay, lane number 2. What's going on in lane number 2? In number 2, we're gonna treat it with BAMH. So over here, if I treat it with BAMH, you're gonna have a cut, as I showed you earlier at the beginning. So you're gonna have 1 small fragment and a bigger fragment. Okay, so the small fragment is gonna be able to go very far. And then you have a longer one that's going to be here. It's not going to be same level as the original because the original is the longest, right? So it has to be below that. Okay, so this is lane 1, lane 2. Okay. What about lane 3? DNA sample treated with BCL1. Okay, so you would take this and then you treat it with BCL1. Okay, so BCL cuts around here, right? So again, you would have 2 fragments. You have 2 fragments. Okay, so lane number 3, you have 2 fragments. And I just want to remind you that the cut is, what is this? A TGA, TCA, right? So it's around here. So this was a little bit longer compared to this one. So maybe it's going to be a little bit higher up, and then like this. Okay, and then for DNA sample treated with both. Okay, so we saw this one is going to be not affected by BCL1. So you will have the same location as before. Okay, and then this is going to get chopped into 2 parts, and it's still relatively small. So we're going to have 2 bands like right there. All right, so that's what you would expect if you run it on the gel. Okay, next one. A linear DNA sequence is treated with 3 different restriction enzymes. The product are loaded into 3 separate lanes, A, B, C. Based on the results, how many times did each restriction enzyme cuts? Okay, so over here, this is markers, or sometimes called like the ladder. It tells you the size of the DNA fragment as a reference. Okay, so like if you get a band here, how do you know how long it is? Well, you compare it to the marker. So that's about 1,000 base pair long. Okay, so here we have linear, right? Linear DNA sequence. And I told you earlier, in linear sequence, the number of cuts will produce n plus 1 number of bands. Okay, so in other words, if you see one band, right, then you got to go backwards now, right? N minus 1. So for lane A, there's no cuts, okay? If you just load the DNA as is, you're just going to have one band. In part B, you see two bands. Two bands equals to one cut. Okay? You take your linear DNA, you cut it once, then you're going to have two bands. And then you have three bands in lane C, right? Three bands, 1, 2, 3 bands. That means it was two cuts. Okay, so if you start with linear sequence, the number of cuts is always going to be one less than the number of bands. Okay, another way to think about it is the number of bands is always going to be one more than the number of cuts. All right, last question, probably the hardest question that we are going to look at. You want to insert a gene into the following plasmid. Okay, so these plasmid, you can buy them nowadays. You just order them online and they will give you a map of the plasmid. So in this plasmid, these are the restriction enzyme site. Okay, so it means if you treat it with E-core-R1, you're gonna cut right here. If you treat it with BMH1, you're gonna cut it right here, so on and so forth. Okay, so ORI means it's the kind of like the promoter. That's where the gene is gonna, that's where the transcription is gonna take begin. Okay, and here we have a AMPR and then a TETR. These are the genes for resistance for ampicillin and for tetracycline. Okay, so explain how you will amplify the gene you are trying to insert. Okay, so that's the first step. You have to clone the gene before you pop it into the plasmid, right? And to clone the gene is easy. We just use PCR. Okay, we just use PCR. Okay, according to the plasmid map, what do these represent? Okay, so I just told you these are recognition sites for various restriction enzyme. Okay, so knowing them, knowing where the sites are, we would be able to know where the gene will be inserted. Okay, so if I have a gene and then it's flanked by the same sequence recognized by E-core-R1, then it's going to go right here. Okay, if it's flanked by sequence the same as BAMH, then it's going to go right here. In fact, if I insert a gene right here, it's going to disrupt the tetracycline resistant gene, right, making it not active. Okay, so I'll explain what that means in a bit. Okay, next one. What are AMPR and TETR? So like I said, the AMPR is a gene that will give resistance to ampicillin. Ampicillin resistant gene. And then the TETR is the tetracycline. Tetracycline resistant gene. Okay, why do they have two of such site? Yeah, how come we don't need, how come we don't just have one antibiotic site? Well, it basically gives you more option. Okay, so remember, in order for us to insert the gene into the plasmid, the gene of interest must be flanked by the same recognition site as as the as shown in the plasmid. Okay, so if I want to insert the gene into equal R1, sorry, into here, then I must have the recognition site on the plasmid and on the gene. Okay, so what was the recognition site for equal R1? Well, it was what was it, like a GAATTC, right? Okay, so you must have this on the gene. Okay, but it's not always possible to have that. Okay, these sequences don't show up, you know, necessarily on all the genes that you're gonna clone. So if you don't have the equal R1 recognition site and you only have the BAMH1 site, okay, then what happens is you will insert the gene right here. So as I mentioned earlier, by inserting the gene into the tetracycline resistant gene, it will disrupt this gene. Okay, so now you won't be able to, even if the bacteria takes up this plasmid, they won't be resistant to tetracycline, because by inserting a gene there, you disrupt this particular gene, right? But fortunately, we still have the ampicillin resistant gene intact, right? So for that reason, by having two resistant gene, it kind of gives you more options as to what you want to do. Okay, so just to summarize, okay, if a gene is inserted into the plasmid using BAMH1, then you will be selecting the bacteria by growing them in ampicillin. Okay, if you insert the gene by treating it with PST1, then you would be selecting the bacteria by growing them in tetracycline. Okay, because inserting it here would disrupt the ampicillin resistant gene while leaving the tetracycline gene intact. But if you insert into the tetracycline gene, you will disrupt the tetracycline gene, but you will leave the ampicillin gene intact. Okay, next question. What is going on here? If you treat the plasma with equal R1, then run it on an agarose gel, how many bands do you see? Okay, so here is our plasmid again. If you treat it with equal R1, you will cut right here, right? So your plasmid will pop open and becomes linear. Okay, so that's just going to be one single band, one band. All right, this is the part that is probably confusing. Let's take a look. If you insert the gene by treating the DNA with BAMH, and then transform it into E. coli, like feeding it, what are the expected results? Okay, so let me just copy this plasmid here, and let's take a look at what's going to happen. Okay. So here it says, if you insert the G by treating your DNA plasmid with BAMH, okay, so I'm going to cut right here, and then I'm going to put my gene basically into this location. Okay, that's my gene of interest. Okay, gonna pop that in. Okay, what's gonna happen in the following? So number one, okay, part A, growth culture with no antibiotics. Okay, so if you put the bacteria in a culture with no antibiotics, then they will grow. So normal growth. Okay, because it's just nutrient broth, right? So they will grow, no problem. Next one, growth culture with tetracycline. Well, do you think they're going to grow or not? If the bacteria did not take up the plasmid, they would die, okay? Because the antibiotic would kill them. But what if they took up the plasmid? Will they survive? Well, even if they take up the plasmid, because we inserted the gene right in the middle of the tetracycline resistant gene, that is going to disrupt the resistant gene for tetracycline. So in other words, even if they take up the plasmid, they won't be able to become resistant to the tetracycline. And as a result, they would all die. So no growth. Okay. If you grow it in ampicillin, right, that's okay, because we've only disrupted the tetracycline gene, but not the ampicillin resistant gene. Okay. So you would expect them to have normal growth. All right. What if this one? Let's see. What if you insert it in the equal R1 location? Okay, so you put the gene of interest right here. That's the gene of interest. Again, no antibiotics. You would expect them to have normal growth. Okay. If you grow them in tetracycline, will they die this time? Well, if they have taken up the plasmid, they will live because the tetracycline gene will be operational. So normal growth. And if you grow them in ampicillin, it's also okay because the gene has not been disrupted. So it's also going to be normal growth. All right. Last question. Predict what will happen in each of the following during gel electrophoresis. If you reverse the electrode, so the current runs from positive to negative, well then DNA samples will run in opposite direction. Opposite direction. So instead of going down the lane, it will be going towards the well. Towards the well. Okay, so if this is your gel, instead of going this way, if you load the sample here, it's gonna go this way and it's just gonna fall off the gel and you're gonna lose the sample. Okay, samples will exit from the top and be lost. Okay. Run the gel for too long, diaphragm is no longer visible. So what is a diaphragm? Well, to give you like a visualization of where the DNA are, you actually add a little bit of a blue dye to it, and the dye makes the density higher, so it sinks into the well. And what happens if the dye usually is going to be running in front of your DNA gel. Okay, so if the dye runs off, like if you see the dye at the edge of the gel, that means your DNA sample is still in the lane. Okay, but if the dye runs off, if all the dye exit at the bottom of the gel, then that means your DNA sample, some of it anyways, it could run off, could also have ran off the gel. Okay, and the sample will also be lost. So DNA sample will run off the gel and be lost. Okay, if you forgot to add ethidium bromide to the stainless solution, what is ethidium bromide? Ethidium bromide is this molecule that will insert themselves into the DNA, and when you shine UV light to it, it will fluoresce. It will give off like a radiation almost, and that allows you to see the DNA on the gel. Okay, so if you forgot to add ethidium bromide, then DNA will not be visible. Will not be visible under UV light. Okay, and then there's no way to visualize the DNA. So that's it. Thank you very much for watching. As usual, if you have any questions, please feel free to contact me.